So it's my pleasure to welcome you to the second part of this symposium. I'm Marie Hudson. As I mentioned, I'm co-director of MI4. I'm going to give you a little bit of a heads up in terms of how we go about setting or preparing for these symposiums. So first of all, we often look back at the evaluations from the year before. So at the end of this meeting, we'll ask you to fill out some of these evaluations because we look at them seriously, and they really help us to craft the next meeting. So we look back, and we thought that long COVID was definitely a priority in the community, so we decided to focus on that and on other sequelae of infections. So we then find a keynote speaker, and we were fortunate to have Professor Iwasaki this afternoon. And then what we tried to do is we tried to get people from within the MI4 community to come and speak around the same subject. And so that's why we're fortunate to have three speakers, what I'd like to call MI4 speakers, who will be presenting in the second part. The other thing, as you've heard us say several times, and it's actually up there, it's really important for us at MI4 not to be identified as lab scientists. We are much more than lab scientists. We have many lab scientists, but we have many clinicians as well, and epidemiologists. And we like to say that we go from molecules to patients to populations. And again, the three speakers this afternoon will be speaking around these three different subjects. The other thing, for those of you who don't know, I'm a rheumatologist, and it's really a pleasure for me to say that two of the speakers this afternoon, Joyce and Brett, have very strong ties to the Division of Rheumatology, and Don and I are doing a project together, so Don, I'm happy to make you an honorary member. So. And just to say, and yesterday I was actually at the American College of Rheumatology, and it's amazing how much rheumatology revolves around microbes, and more, and there are so many exciting opportunities out there. So basically what I'm implying is rheumatology is about to take over MI4. But having said that, let's be a little bit more serious. So the first person I'm going to introduce is Joyce Rauch. I've known Joyce for many, many years. She's a professor of medicine and a longtime member of the Division of Rheumatology and Experimental Medicine. She's also, I was surprised to read, an associate member of the Department of MIM, so that's fantastic. Joyce received her PhD from McGill and then completed her postdoctoral training at Tufts. She established her lab at McGill over 40 years ago. That's fantastic as well. And her research focuses on mechanistic roles of cell death and innate and adaptive immunity in the initiation and pathogenesis of autoimmune disease, especially systemic lupus erythematosus, but her other main area of research is antiphospholipid syndrome. And so along with Judith Mandel, who's actually here today, they were funded together with this sort of idea of creating new teams to study the impact of viral infection on the initiation and development of systemic lupus erythematosus. So Joyce will be representing the Rauch-Mandel team. So welcome, Joyce.
Thanks so much, Marie, for that introduction, despite the age-related information. But really, it's a pleasure to be here today and an honor to speak. You might want to rename this session after you hear me, from mice to patients to populations. But yeah, we're not quite at the molecules yet, but we're starting. So this was really a pilot project, so I want to warn you about. So let me go ahead. So as Marie said, I'm going to talk about the impact of viral infection on the initiation and progression of systemic lupus erythematosus, which we shorten as SLE. And this is a collaboration between my laboratory and that of Dr. Judith Mandel, and it was funded by a seed fund grant from MI4. So SLE is a systemic autoimmune disease characterized by involvement of multiple organs. And you can see in the slide that goes anywhere from the skin and hair to the heart, to kidneys, joints, and lungs, just to mention a few. The natural history of SLE shows a kind of relapsing, remitting course. And what I want to highlight here are the peaks with flames and the activity periods or flares. and the valleys, which represent the inactivity or less active periods. But throughout, there's usually an accrual of disease damage in the patient. So the origin and progression of SLE is very complex and multifactorial, but we know two things. And we know that genetics and environmental factors play key roles. This slide from Tsokos in the New England Review divides SLE into pre-SLE, which is asymptomatic, followed by an early SLE phase where there are autoantibodies but no symptoms, and then the SLE diagnosis phase where there are symptoms that manifest themselves. I'm going to talk today about viruses as being one of the potential environmental triggers, both in the early phase and in the later phase of SLE. So viral infections, we believe, can be involved both in the initiation and the progression of SLE. So there is evidence in the literature of increased risk of hospitalization for SLE flares following influenza infection, and this was an article by Zhu et al. In 2021. This was a self-controlled case series design. So what that means is that the patients themselves acted as their own controls. The risk interval, sorry, didn't mean to show that. The risk interval was considered, I don't have a pointer, but the 7 days following the diagnosis of influenza infection. And the control intervals were the weeks surrounding that period. So the whole year surrounding that period were the control intervals. And what this group found was during the risk interval for 7 days of infection versus the control interval, there was a higher incidence of hospitalization for SLE flares. And the incidence ratio was 25.75. Interestingly, that was most evident in the first 3 days post-infection. So what creates this potential link between viral infections and SLE? Well, one potential scenario is because viral infection induces the production of type 1 interference at a very high level. We know SLE is also or type 1 interference are also associated with SLE. And some of the evidence pointing to that is that type 1 interferon serum levels are associated with SLE disease activity. And cells from SLE patients have a type 1 interferon stimulated gene signature. Also, in the early days, when type 1 interferon was used as a treatment in non-autoimmune individuals, there were manifestations of SLE transiently in some of these individuals. And I'll add to that that anti-interferon, type 1 interferons are also being used today in treating SLE. So we hypothesized, based on all I've told you, that viruses play a key role in the development of SLE, and that one potential mechanism is through type 1 interferon production and signaling. So our research questions specifically were, does viral infection trigger the initiation of SLE? And the second is, does viral infection accelerate the progression of SLE? And the experimental approach we used was an induced model of SLE, where both the initiation of the disease and the viral trigger can be controlled. So our induced model of murine SLE involves the immunization of mice with human beta-2 glycoprotein 1, I'll call beta-2 GP1. It's a plasma protein that's present in all of us, a very normal protein, but it's targeted in SLE by autoantibodies, and LPS, which was used to stimulate an innate immune response. We usually repeat immunization every 14 days for usually 4 immunizations, and bleed after each immunization, about 10 days after, and then we assay for serum autoantibodies. And the autoantibodies are SLE-related or SLE-specific autoantibodies I've listed there. And what I'd like you to note here is how these autoantibodies develop in our model. So not surprisingly, when we immunize, and this is old data published in 2006 where we described the model, antibodies to the immunizing antigen are very high after the first immunization. Antibodies to related antigen, cardiolipin, which binds beta-2, are also high. But what surprised us very much after we immunized the second time was we started to see SLE-related and SLE-specific autoantibodies, such as anti-DNA, double-stranded DNA, anti-LA, and anti-Rho. And then when we immunized further, we saw even more SLE-specific antibodies, such as anti-SM and anti-NRMP. And this excited us a lot because it replicated what was seen, what has been seen in human lupus. And this is a figure from Arbuckle and coworkers from 2003, where they looked at individuals 5 years and more before they were diagnosed for SLE. And they found that many of these individuals had significant levels of autoantibodies up to 5 years or more pre-diagnosis. So using our induced model of SLE, we asked, does viral infection trigger the initiation of SLE? And the virus we chose was LCMV, the Armstrong strain. The reason we chose this was it shows an acute viral infection in mice. The virus titer peaks at 2 days after infection, and the infected mice show no sign of illness, and the virus is cleared from the organs about 10 to 14 days after infection. This little insert just shows you the natural course of LCMV Armstrong infection, and I want you to note that type 1 interferon production peaks at 2 days. So we used this information to design our initial experiments, and we timed our studies the following way. We thought the following rationale was, let's look at the peak interferon production at the time of initial antigen presentation. So we infected 2 days prior to the antigen administration. And then the second experiment looked at peak interferon at the time, or we timed it to look at peak interferon at the time of germinal center reaction, so LCMV 6 days after immunization. And then the third time point was to try to time the type 1 interferon at the time of autoantibody production, so 14 days after immunization. And that just shows you the immunization with beta-2 LPS to induce the SLE model, and that was done twice in the study. So the treatment groups are listed here, but I'm going to repeat them in other slides, so I will focus on them there. So you see the same schematic, and this was, as the title says, does LCMV, to ask the question, does LCMV prior to SLE initiation impact autoantibody production? So we're giving LCMV 2 days before, and what I will do is go through the treatment groups with you. The next 4 slides are going to look very similar with the same sort of antibodies, so I'm going to go through them very quickly just because we have 15-minute time period. So the first treatment was LCMV alone, as you see with the arrow, and that's you can see just across the baseline, there was no antibody induction. The second line you see is open circles and a black line, and that's our SLE model induced. And you can see that there's a nice anti-beta-2 glycoprotein 1 antibody response. And then the third treatment group was the SLE model with LCMV infection. And what I'd like you to note here is actually the LCMV infected group was lower than the SLE model alone, which surprised us, but this was what we saw with this particular time point. And this replicated with the other autoantibodies, although it was most evident with anti-beta-2. So we conclude from here that LCMV prior to SLE induction appears to diminish or minimally affect SLE autoantibody production. So we then went to our next time point, which was 6 days following SLE initiation. So it's the same sort of design, and I'm going to show you the same type of antibodies, but I want you to focus on the results. So LCMV alone shows really nothing, no autoantibodies. The beta-2 plus LPS, the SLE model, shows a nice antibody profile. And then the beta-2 plus LPS and the LCMV, so our SLE-induced model and LCMV, is here showing a higher level of antibody production. And this is being replicated throughout these 4 different antibodies, which I don't think I mentioned to you, are anti-beta-2, glycoprotein 1, anti-cardiolipin, anti-double-stranded DNA, and anti-Rho. And we've assayed the other antibodies I mentioned earlier on, but I don't have time to show them, and they all show the same tendency. So the conclusion for this particular time point was LCMV infection 6 days after SLE initiation enhances autoantibody production. The third time point we looked at was 14 days following SLE initiation. So it's the same sort of setup. The only thing, I apologize, the LCMV group is in pink here. And this was a little less interesting, but shows an enhanced response, but mostly toward the end of the period we were looking at. So here we conclude that the LCMV infection 14 days after SLE initiation appears to enhance autoantibody production, but a longer time course is really needed to verify this effect. So the summary basically is LCMV infection enhanced SLE autoantibody production, particularly 6 days after initiation. But what was our next question? Well, how is this happening? What is the mechanism responsible for the effect on the autoantibody response? And not surprisingly, we wanted to look first at type 1 interferons and whether they were responsible. So again, this is a very similar experiment. The only thing that differs here is we used a mouse that does not have a receptor for type 1 interferons, so cannot respond to type 1 interferons. And we did the same sort of treatment groups, the SLE model alone and the SLE model with LCMV 6 days post second immunization. And what I want you to note here, again, is the SLE group alone with the black and white arrow, and the SLE induction group with LCMV in the blue arrow. And for virtually all of these, we are seeing a higher response with LCMV. I'd say anti-Rho is questionable, but for the others, we're seeing a higher response. So what does this mean? Well, this shows the IFNOR deficient, so the receptor deficient mice infected with LCMV following SLE initiation have increased autoantibody levels despite not being able to respond to type 1 interferons, suggesting that interferon 1 may not be required for the LCMV effect. So just to summarize all the data, using an induced murine model of SLE and LCMV as our viral model, we found that LCMV infection after SLE initiation resulted in enhanced autoantibody production. The enhanced autoantibody production does not seem to be dependent on type 1 interferons, suggesting there might be a type 1 interferon-independent effect of the virus. LCMV affected the autoantibody response early in the initiation of autoantibody production, so 6 days post immunization, and experiments are ongoing to evaluate the impact of LCMV on immune cells, particularly B and T cells from the germinal center. These early findings provide a proof of concept that virus can impact SLE initiation, at least in mice, and a model in which both SLE initiation and the viral trigger can be controlled and the cellular and molecular mechanisms investigated. Future directions, and these just kind of are repetitive, so we're going to look at mechanisms by which LCMV impacts initiation of SLE, and I've said what is written there. We're also going to look at how LCMV infection impacts the progression of SLE, and we'll investigate whether other viruses such as influenza can impact the initiation and progression. And the acknowledgement slide is the most important. These are the people who did the work. So in my laboratory, Celeste Pilon, who's in the audience, was really the project leader, thought of the idea, and has been instrumental throughout. Aishwarya Skaria and Elena particularly Anina, who have done the work on the assays, and Elena some immunization. In the Mandel lab, Dr. Mandel, of course, and Dakota Rogers, a PhD student in her lab, my close collaborator, Jerry Levine, and special mentions to Dr. Piccirillo and his laboratory members for support and advice, and Goodman Cancer Center, the animal facility and immunophenotyping platform. And without the funding, last but not least, without the funding of MI4, this would not have gotten off the ground. So MI4 and CIHR for the independent grants to Dr. Mendel's and my own laboratory to supplement the experiments where we needed it. There is a new and upcoming grant I'm excited to report from Lupus Foundation, special interest grant. And thank you for listening and questions. Hopefully I didn't overstep my time.
I think we have time for one question. If there's any pressing questions, I have some, but I'll save them for later. Okay. So Ara.
Thanks, Joyce. So do these mice develop SLE symptoms?
So by symptoms, you mean they develop autoantibodies, and they develop, our initial study showed the development of glomerulonephritis, very much like SLE, renal disease.
And do they exhibit sickness behavior?
We didn't, did they exhibit sickness, you say?
Yeah.
Not that we did not, not that we could detect. We didn't study cognitive behavior. We didn't see any dermal, any skin manifestations. So other than autoantibodies and renal, that was really those were the major findings.
I'm just wondering if you take it out and let these mice, go for a while, do they eventually develop these more additional evidence of SLE that the virus may be influencing beyond autoantibodies?
Oh, so you're talking about the virus? Yeah, I'm talking about our model, just our model. We haven't taken the mice infected with virus out much further.
Yeah, that might
In fact, we
that
cut
might be
those.
informative. That might be informative.
That might be very informative because, as you see, they seem to be keeping the level of autoantibody up, and we really need to take them out further. These are really pilot studies, very early on, a year and a half into a new study. Thanks. So I guess that's it.
Yeah,
Thank you. Anyone can just. Thank you.
Thank you for your contribution. Thanks. All right. So moving on, our next speaker is Don Vin. I don't think that Don needs much of a very long introduction. He is a valued member of the MI4 community, not only for his science, but also for his advocacy during COVID, his ability to represent MI4, the community, and his work with the lay public. I will supplement that by saying that Don, as many of you know, is an infectious disease physician, microbiologist, and section head of the ID Hematology Oncology Transplant at the MUHC. He is an FRQS senior clinician scientist, and his research program focuses on deciphering how molecular defects of human immunity contribute to infections that are recurrent, recalcitrant, or severe. So he's going to be presenting some of his work today. The title of his talk is Candida. We'll bring that up shortly. Candida, Card9, and Cognition. So thank you, Don.
Thank you very much, Marie. So as we get that set up, I just want to thank the MI4 Executive Committee and Secretariat for the opportunity to present. I was told, can you talk about complications of an acute event? So I said, well, sure, I can actually render homage to Marcel and do my version of Billy Joel's Piano Man, but I thought those complications would be both irreversible and potentially debilitating. So I will instead talk about my science, which isn't this. So I am hoping that it will. Joyce, out of curiosity, I don't know your slide deck. Do I just keep going? I don't know. This isn't my slide deck at all. But if anybody wants to hear about LCMV.
They were extra. Just get out of it.
Oh, okay.
They were extra. Sorry.
No problem. Do I close and then I open mine, or how does that work?
I'm not sure where yours is. This one?
I hope my 15 minutes hasn't started. Okay, so I was, we've heard a lot about COVID, so I'm certainly not going to talk about that big C, but I want to talk about the other C's that are going on in our lab, which is related to Candida. So here I am, and I just want to talk to you first about the context, which is that, I'm very fortunate as a clinician scientist that my lab is attached to the hospital. And what that allows me to do is that allows me to study patients. And they're relatively captive in our four walls of our hospital. And I'm particularly interested in infections, as mentioned by Marie, that are recurrent, refractory to treatment, or severe. But why that's important is because I can not only just take care of them or try to take care of them while they're in the hospital, but I can try to formulate the problems that come to mind when we evaluate them. Why do they have this infection? Why are they not responding to treatment? What can we do to improve it? And we can answer these questions, where we can pose these questions directly on these patients, on individual patients, and then we can study them. We can take their blood samples or their biological samples and bring it to my lab, and in my lab, we can then use a variety of approaches that are both genomic, molecular biology, and immunology to try and answer the question of the patient that lies before us. And of course, as a clinician, you want to close the loop because you want to be able to take that information that you identify in your research lab and figure out some way to help the patient. And in this manner, we sort of follow the guiding principle of Henry Kunkel, a very famous Rockefeller scientist, whose motto was, study a few patients really well. So when we talk about infection, we have to, and for this symposium, the consequences of infection, we're really talking about, first and foremost, an acute infection, right? As an infectious disease person, a physician, that's what I see. So when you talk about an acute infection, what usually happens is that there is if you look at disease trajectory, there's a peak in symptomatology, and then if you do things properly, or maybe if you do nothing and the immune system kicks in, it should resolve. We often get involved when it doesn't resolve, when it either persists or progresses, or there's also a possibility where the disease manifests, it resolves, and then there's a recrudescence. Now, all of this is in the context of an infectious disease. They start off with an acute infection, and what either resolves, persists, progresses, or recrudesces is the infectious disease. Usually it's the same symptomatology or somewhat related to the acute episode. But we do know, in terms of complications, that acute infections can lead to novel or neophenotypes. Here is just a bit of a sample of some slides that I put together of infections that are known to do that, right? So even though SARS-CoV-2 can cause both an acute respiratory infection and long COVID, it is by no means the only infection that can do that. Some of these phenotypes are so profoundly different than the original infectious episode that it still remains enigmatic as to how that happens. And those examples are listed here. I'll give you some examples of what we're working on in our lab. So I just want to present to you patient 1, P1, who is a French-Canadian male who presented at the age of 30 with seizures while playing hockey, and over the ensuing 6 years had recurrent seizures. The investigations for his seizures showed this, like what we call an unidentified bright object, or UBO, on his that's his brain. So again, just to reorient you, that big white thing is not normal. He was told that this was cancer or some sort of tumorous mass, and eventually, because of his recurrent seizures, underwent a resection. And there was no mass, there was no tumor, there was no cancer. It was pure Candida albicans, which is a yeast. The same yeast that causes diaper rash or vaginitis, except that in his case, it was in his head. We also had a second patient, P2, who was also French-Canadian, who presented with recurrent seizures. And on imaging, he had those little bubbles that I can't really show you with a pointer, but trust me, those bubbles are not supposed to be there. And those bubbles were actually said to be glioblastoma, which is a type of cancer. But again, on resection, there was no cancer. It was pure Candida albicans. Both of these individuals were put on the appropriate antifungal therapy, and despite that, they had recurrent seizures and an ongoing disease, which I'll characterize in a second. We also had another patient, P3. She actually presented with trouble with her vision, went to her optometrist, who sent her quickly to the emergency room because they saw this big white floating ball. I don't think you can appreciate that, but like none of us have floating cotton balls in our eyes, okay? So that was abnormal in her. And then when we were contacted, we said, can you scan her head and see what's going on there? And she happened to have this bubble configuration in her brain as well. And she also had back pain, for which we imaged her, and she had an infection in her spine. She had a twin sister, and her twin sister was relatively asymptomatic or polysymptomatic, but when we went looking for disease, you could see that she had scar formations in her brain. I don't know if I can show you, but I don't want to risk it. But anyways, all that is, you can see in the cerebellum, the bottom right, there's like a white scar tissue in her brain. So first of all, we put a term to this diagnosis, to this disease. We called it spontaneous central nervous system candidiasis, or SCNSC. And what we did is we said, in the absence of an obvious extrinsic immunological compromising condition, they must have an inborn immunocompromising condition. So we sequenced them, and long story short is we found a mutation in a gene called CARD9. The first patient was homozygous for this variant that changes the tyrosine to position 91 to histidine. P2, P3, and P4 were compound heterozygous, so they carried the same Y91H mutation as P1, and then they had a second mutation, which we're going to loosely call a regulatory mutation. And what that means is if you take otherwise healthy people and you look at the genomic DNA at this location, you can see, the wild type sequence. Which is listed there as a T. And if you were to see how much of that, or what does that gene express, well, in wild type healthy controls, the T at that location at the genomic DNA level is also expressed, that T is expressed at the mRNA or cDNA level. If you take P2, what you see is that the genomic DNA level, he's heterozygous for this PY91H mutation, but the disease that he has, the syndrome, tells us that he has something pathognomonic for CARD9 deficiency. And so when you look at the cDNA level, you see that it is predominantly, if not exclusively, the mutant allele that's expressed. Same thing with P3. Heterozygous at the genomic DNA level, homozygous, if you will, at the transcript level. Same thing with her twin sister, who is heterozygous at the genomic DNA level, but homozygous. And we know that if you take their mother, and you see who is completely asymptomatic, she's heterozygous at the genomic DNA level and heterozygous at the cDNA level. In other words, under normal conditions, both alleles are expressed, and in P2, P3, and P4, they are not. There is an allelic imbalance. Only the mutant allele is expressed. So these 4 individuals are transcriptionally equivalent to each other from a CARD9 mutation perspective. We were able to study their immunological response to C. albicans in vitro. Mathieu Albert and the Milieu Interieur Consortium at the time had this very comprehensive immunological investigations that showed particularly what's here in the red box, which is that otherwise healthy controls, if you take their PBMCs and you hit them with a variety of agonists, if you hit them with heat-killed Candida albicans, there is a distinct production of GM-CSF. And when you look in our patients, well, what you can see here is P1 and P2. If you take their PBMCs and you stimulate them with C. albicans or things that are either commercial or look like C. albicans, well, in fact, they do not produce GM-CSF. So that balance between what healthy controls normally express and what these patients do not express to C. albicans, we think gives us an idea of how humans respond to C. albicans. And so we were able to translate that information. This is a bunch of lumbar punctures or spinal taps on P1. What you can see is perhaps, you can't see, but I can, which is that there is a dotted line above the 50, which is supposed to be in blue, and that means that if you're above that dotted line, if your blue curve is above that dotted line, it's abnormal. And below, there's a black dotted line, which means that if your black curve is above that dotted line, it's also abnormal. So what we did here is we measured the amount of cells in the cerebrospinal fluid of P1 or the protein, either in black or blue respectively, and we mapped it over time. And as you can see, he's grossly abnormal. There were times where it is indicated by a red cross, you can even grow Candida albicans out of cerebrospinal fluid. And we tried all sorts of the right antifungals listed on top. You don't need to worry about those, but we tried all sorts of antifungals which should have worked, but didn't. You can keep isolating Candida albicans out of his cerebrospinal fluid, and his parameters of the cerebrospinal fluid remain continuously abnormal until we hit him with GM-CSF. If he was unable to produce GM-CSF, could we just give him GM-CSF, which is licensed to use in humans for other reasons? And would it make him better? And the answer is, it looks like it based on his CSF parameters. And of course, when we made the mistake of stopping it, well, then he got worse. And then we gave it to him again, and then he got better, to the point where he was able to function as an engineer. He initially actually was an engineer for SNC-Lavalin, and then got to his senses when his brain started working again, and then worked for another company, and avoided a lot of issues. Now, you can say, well, 1 point is lucky, and 2 points is a trend. So this is P2, and we did the exact same thing with P2. Remember, he had the bubbles in his brain. He had the resection of those lesions. And at this point, he's 39 years old, and he was told he was palliative because there's nothing to do for his ongoing seizures. This was as good as it got from their perspective. We, again, showed that he had impaired production of GM-CSF. We gave him GM-CSF, and several years later, he remains not only alive and semi-autonomous, his brain looks like it's healed. He's actually lost his palliative care privileges. So now, how do we take that information a little bit further? Well, what we were able to do, and I don't know if Maria Landekaj is here, but she was a PhD student in my lab, and she was at the forefront of helping us study what's going on, help us study card9 deficiency using a mouse. And what we were able to say is, first of all, you know, when you talk about a mouse model, we actually are using the word model in its actual sense, which is that it is a representation of a disease that we cannot otherwise study in natural space, right? We want to be able to have a model that phenocopies or recapitulates our human disease and then do things on that mouse that we can't do on humans. So the original studies on card9, this is before us, showed that compared card9 wild-type mice to those who were null, they infected them with a high dose of Candida albicans. All the mice died. The null mice died sooner than the wild-type mice. And long story short, card9 was important for antifungal immunity. That was groundbreaking. But that's not what we want to do. We wanted to study card9 deficiency. So instead of going for discovery, we wanted to study immunopathogenesis. We want to study disease. How does this card9 loss of function lead to disease? And so what which we ended up doing with collaboration from Sal Qureshi and many others in this lab, including Mazdi Vangahi, is we created a mouse model of this Y91H mutation. And then instead of titrating the Candida albicans dose to the wild type, and then seeing, you know, how sooner the mutant mice died, we actually titrated to the card9 genotype because that's what we were interested in. And we were able to find a dose where the card9 mutant mice died and the wild type mice were completely unaffected. And I'm going to try and show you a video. This is my first time ever showing a video, and I'm hoping it works. Now, these 4 mice are all card9 Y91 knock-in mice. And what you're going to see is that there's 2 mice that are moving around well, bright-eyed, active, and reactive, as we call it, bar. And then there's 2 other ones that aren't. One a little bit less so than the others. He's a little bit doing his own thing there in the corner, which is, of course, abnormal. And in fact, there was a whole range of neurological symptoms associated with these people. How does this thing stop? So that's, okay, let's just fast forward here. Anyways, all that to say is that we were able to create a knock-in mouse model that recapitulated the neurological symptoms. Histologically, what we were able to show is that these card9 mutant mice had these brain lesions that are not seen in wild type. When you do the histology, what you see is that they have multinucleated giant cells, which, by the way, we're able to go back to our human data because we had brain biopsies on the humans, and they also had multinucleated giant cells. So you're talking about neurological symptoms, you're talking about histological resemblance, and last but not least, one of our mice, or some of our mice, had what we call skull deformation, where because of the infection, their skulls just cracked. And we were able to identify that on autopsy. And in fact, if you go back to the original description of card9 deficiency, severe skull destruction was actually one of the symptoms of this disease. So what can we do with this mouse model? Well, as an infectious disease person, we want to take these card9 deficient mice, this model that we've developed, and we want to be able to understand how this card9 loss of function leads to invasive candidiasis and spontaneous central nervous system candidiasis. And of course, we want to understand how does GM-CSF account for the benefit that we see clinically. But there's actually a different story that should be also prodded. What more can we do with this mouse model? Well, there's this growing body of literature that suggests that in humans who have Alzheimer's disease and you do autopsies on their brains, you can actually find evidence of fungal infection. Initially, it was Candida. I've become a little bit more skeptic because they've found also different sorts of fungi, and they always happen to be 100% in the Alzheimer's and 0 in the controls. But nonetheless, it looks like there's maybe a signal that Candida could play a role in Alzheimer's disease. And in a different group, David Corey's group, actually showed that in the mouse model, what happens when you infect mice with Candida albicans at low dose is that you get this amyloid precursor protein that then triggers the microglia and can lead to production of an amyloid beta-like peptide. So we think that in some cases where you have fungal-associated Alzheimer's disease, we can use Candida, not Candy, I don't know who she is, but Candida, and use that as an overlap with card9 deficiency, so we can use our mouse model to study Alzheimer's disease. And just to let you know, just one last thing, which is that in Alzheimer's disease, they're actually using GM-CSF as a treatment option. Because in the mouse model of Alzheimer's disease, GM-CSF does a lot of things to improve the mouse model of Alzheimer's disease, which I won't have time to get into here. But in the human model of that, GM-CSF therapy, small little studies, but you seem to have an improvement in cognitive function compared to baseline or compared to the placebo arm. That cognitive function improved well into over a month and a half. And plasma markers of Alzheimer's disease also improved with GM-CSF. So all that to say is by studying a few patients well, I think we can understand not only the human disease of interest, maybe even tangential human diseases of interest. Again, we study human diseases, we call that medicine, and if you study animal diseases, we call that veterinary medicine. If you study animals in lab conditions to understand human diseases, you're a scientist, but I think we can't overlook the fact that studying humans in a natural condition to understand human diseases is also important. We started off with card9 deficiency, talking about SC and SC, and we're going to continue to characterize the disease. We're going to use our mouse models to understand it, but we also want to perhaps use this model to understand other neurodegenerative diseases. There's a lot of people here I have to thank, and they're all listed here. Thank you very much.
Again, we have time for a burning question. Marcel?
Hi, Don. If I understand, most of your patients were men?
No, so 2 of them were men, 2 of them were women, and then we've had other ones that had a different fungus who have also been about 1-to-1.
Okay, about a 1-to-1 ratio. And what do you think the route of infection in the men is? And how do you model that in mice?
So we think all of the routes of infection for invasive candidiasis, whether it's this or other, is through the gut. Yeah. And so in fact, when you infect the newborn pup, if you will, within a 1-week-old mouse pup, in the absence of any chemotherapy or steroids that you would normally use for the adult mouse, you can actually, and you gavage them at high dose, you can actually get invasive disease.
Okay, thanks.
Yeah.
Okay. Thank you very much. I will move this out of the way. So, and perhaps... So last but not least, I'd like to introduce Brett Toombs. So I've known Brett for many years, ever since he joined McGill, I think in 2006. I will not tell you about the first time we played curling together. You can ask him about that. We'll stick to the topic. So Brett is a psychologist. He's a fantastic collaborator. He also has a great interest in scleroderma, which is something that's very dear to my heart, and that's how we met. Brett trained in the US, did his postdoctoral fellowship at Hopkins, and then came to McGill in 2006. He's presently professor and Tier 1 Canada Research Chair in the Department of Psychiatry. Among his many accomplishments, and again, I will not name them all, he founded this international cohort that's called the Scleroderma Patient Intervention Network, so SPIN, which is really an international group of people, investigators, patients, doing some really innovative research and trying to do psychosocial and rehab interventions in a rare disease, and really leading the field in this area. He leveraged this large international cohort to study mental health, and we've heard a little bit of mental health earlier today, in this at-risk population of patients with scleroderma during the pandemic. And he did publish some surprising results. Today he's going to talk to us about mental health and COVID-19 evidence from a systematic living systematic review, but I hope that you will address some of your more controversial work as well. Thank you, Brett.
Okay, thanks, Marie. First of all, we don't have any controversial work. We're always right. No, I'm just kidding. But I've had a tough 30 minutes or so, Marie, because when you said that Dr. Rauch has founded her lab for 40 years ago, I was worried that nobody could possibly take me seriously. It's been less than 40 years, but so I wanted to announce to everybody that I found out this week that I can now go to the senior swim sweat session at my local swimming pool. So you can take this seriously, I think, hopefully. I want to point out that I'll be talking about mental health, and I should have a slide on this, but I want to differentiate that from having a bad day or being bored or being angry, or because mental health means that your mind is not in a place where you can do what you need to do, you can do what you want to do, you can enjoy something in life. It doesn't mean that you're happy all the time. So there's been a lot of confusion about that. So we're talking about mental health and COVID-19. And one thing that we do, so apart from our scleroderma work, I have another part of my team that does kind of big data. So we look at mental health assessment and diagnosis. We put together, we collect data from investigators around the world, and we probably have 350 data sets from primary studies we bring in, and we pull them all together. So we have a lot of expertise in processing data at large amounts of data at a fairly fast rate, which was critical here, because if you think back naively, I think we were at least worried at the beginning of COVID if we'd have enough data, how long was it going to last? And we turned out to have the opposite problem. What do you do with all these data out there? How could you possibly make sense of any of them? So anyway, we've systematically reviewed what was going on in COVID on mental health. So just a quick disclosure and also thank you. So we got this project off the ground with a seed grant from MI4 very quickly. MI4 got into action very fast and got us going into COVID along with many other people. And then we've since funded this project with a number of grants from CHR. But the message that got out in COVID, and this term got used a lot, that there was a mental health tsunami. It was a preordained or pre-understood that the mental health in the population was crashing. And if you look back here, and we have looked back and looked at this, experts at the beginning of the pandemic, before there were data, were saying quite factually that the mental health of the population was collapsing. They began to say that people were committing suicide, that it was all unraveling. And I should say also, as I going forward, by the way, that some people did have a terrible time with COVID. So and people have terrible times now. Today, there's people out there suffering with mental health. So we're, of course, talking about the aggregate, what happens at a population level generally. But these kind of things were all coming out, and there were stronger statements in the newspapers, and our experts were out there running around telling people what was happening before they had any data. And it was all quite negative. They were telling people they weren't coping, that they were falling apart. And we kind of saw this coming on, so we decided that we wanted to try to go out and find a way to get data to answer this question. What's actually happening? What does the evidence actually tell us, right? And some doubts later on began to be put forward if it really was the case, right, that there was a tsunami in mental health, that people weren't able to cope with what was happening in their lives. And a lot of evidence began to put forward suggesting that mental health was going very badly. And a lot of evidence was getting published. And we've all seen, we could go on for a long time about the, thank you, Marcel, about the quality of COVID. I think there's been probably now at least 500 retracted papers and other sloppiness, but a lot of mental health research began to be published that said, oh, oops, mental health is going badly, this is terrible. But if you think, these were all virtually all cross-sectional studies. People would pass out a survey to people at one point in time, and they'd answer questions, and they'd say, oh, this looks very bad. And then they'd proclaim in an article that this was very bad. But our surveys in mental health aren't designed for that. And just to give a quick methods on what happens here, people have all seen these questionnaires we give out. There's any number of them. They ask you a number of questions. Each item has points to it. They add them up and you have a score for your mental health. And the way these are calibrated, because at some point you've got to put a line and say, oh, okay, over this line is the bad point, and over below this line is not. And the way these are actually set up, they're set up for screening. They're set up where, if you give a score, you basically have you interview people, you have a diagnosis, you figure out who actually has, say, depression, who doesn't. You give a questionnaire, you move this threshold up and down, you feel, okay, this threshold is a good point to screen. This catches a lot of people. And so we have lines like that on our questionnaires, but they're not meant to tell you that. Imagine doing that with a mammography and reporting that as your breast cancer prevalence, right? We don't do that. Now that would be even more extreme because the rates on the test, but that's kind of what goes on here. But as we got, all along, it's gotten, I guess over the probably 20 years or so that I've been doing this, increasingly people are using these questionnaires and acting as if they're like diagnoses, acting as if mammography means you have breast cancer. And we in our other project I mentioned earlier, we collect these large data sets, so we look at 3 different commonly used tools in depression. The PHQ-9 is used in general mental health, or the Hospital Anxiety and Depression Scale, the HADS, is used in hospitals, and the EPDS is used in perinatal care. But we compared what happens when you use these things to say how many people have depression compared to actual diagnoses, and they usually double or triple the actual rates in any time. okay? And there's literally dozens or hundreds of different tools and questionnaires that people use. So if you imagine if you go out there and you do this cross-sectionally and you look at this, you could get a high rate just on the false positives alone. And so this, if you do it cross-sectionally, you know ahead of time you're going to have a story that says this looks bad. Okay, in essence, it's bad science. And these were being cranked out of our, imagine, what is that phrase they say, if you have a hammer, everything looks like a nail. Imagine every time you got a mask, they were also giving you these hammers. And everyone was running out and publishing mental health research cross-sectionally and saying, oh, this looks really bad. But, and what do you get from this? You start putting this together and you start getting things like this. I could have found many other reviews that said 12 to 87% of people have depression. Well, what does that even mean? Okay, so we set up to do this review, and what we did is it's very simple in some ways. The processing the amount of data we ended up having to process was very complicated. But in the end, we said, well, if you want to know if mental health has changed, you need to look at where it was before and where it is now. It's very, very simple. It could be, there's some complexity here because people report higher mental health levels over the years. Over the last decade or two, what they're reporting is going up. Now, we don't know if that means that they're reporting differently than they used to because of how we think about it, or if it's actually going up and people actually feel worse. That's a really tough question. But it's going up slightly over the last couple of decades. But in any case, if we want to know grossly what's happening, you need to look what's happening before and after. So we did that, and the team has been incredible. But we went through, and we've looked at some other things. We're looking at some interventional work and some other things in the project. But this part of this we just compared if we found any cohort that was following the same group of people before the pandemic within 2 years. And it can't be back 10 years ago when you had people that were in their childhood and now are young adults because of the developmental differences. But we wanted to say, within the last 2 years prior to the pandemic to sometime during the pandemic, what happened to mental health symptoms? So very simple question. They had to compare basically the same group of participants or use some reasonable statistics to make up for people who dropped out along the way. And we found at that point, this came out in last March in BMJ, our paper. So this was from early in the pandemic. So we mostly had data from the first, say, 6 months of the pandemic here. And how do we figure out if things have changed? I mentioned a few minutes ago that people use dozens or hundreds of different measures, right? But in the end, if you have a measure of depressive symptoms, they're not really that different from each other in what they report. They correlate very, very highly with each other. Probably 0.8, 0.85 at any given time. They're not that different. So what we do, but if you have a score on one that has a maximum score of 10, and a score on another measure or questionnaire that has a maximum score of 60, you can't just say one is a 55 and one has an 8. So what do we do with that? Some fairly basic statistics. We put them in terms of standard deviations. Okay, so how many standard deviations do these things change across the pandemic? And what does this represent here? If you look at the purple graphic is like it's a normal curve. That could be the pre-pandemic distribution of scores. And what we say is that if they move, the yellow curve is a post-pandemic curve, and that's about a quarter of a standard deviation. That's, by the way, about how much you get from treating depression, major depression. So on average, but in any case, you move the distribution about that much. So that's about a quarter of a standard deviation. So we took each of these cohorts, and we did a number of things. We evaluated, we rated the risk of bias or the quality of the studies that were done. But we coded them in terms of how many standard deviations they've changed, either going up or going down, from before the pandemic to during the pandemic. And we looked at, we broke them up in a number of subgroups. So we looked at, you'll see here, general population. We did it by sex or gender, older adults, young adults, university students, and other populations. We didn't have a lot of studies from low and middle income countries at that point. From some other important groups, racialized minorities, we didn't. People weren't publishing that. But you'll see here, if you look here, this was for general mental health, which is basically a concept that blends kind of anxiety and depression together. And you'll see here that the ones that are shaded were statistically significant, but if you think about the graphic of a quarter of a standard deviation from the slide before, they were all pretty close to zero change here across any of these groups. And some of these were pretty large. The general population, we had about 30,000 participants. And one thing that was notable about this research is that there weren't many really, should be kind of a wake-up call to Canada too, that we don't have very many good general population cohorts that track this. I think the Dutch had a great one, the British had one, some that would follow the general population going forward and had really good data on their populations. And they were pretty consistent with this, zero or small changes. But we don't really have those going on here that we're reporting quickly in Canada. This was for anxiety symptoms, and you'll see it's about the same. It didn't change very much. This was for symptoms of depression. There's more gray areas here, but again, they're still very small. And I think if you put this in perspective, this probably means that there's some change going on in some of the population groups. It's on average, this level is something you probably wouldn't experience, or if it were you, it's a difference, but it's a small difference. And kind of in summary, you see that the one group that we found across, we couldn't disassociate sex from gender just because studies didn't report it that way, and we could only do what studies report in their publications. But for females or women, there was a small amount of change that was consistent, whether we look at general mental health, anxiety, depression. But nothing else was consistently changed at all. And there were a couple that were actually even positive, that people with pre-existing mental health conditions, these were very small, but they actually got a little bit better. And we have a patient that works with us all the time who has serious mental illness, and I think it's something that this is actually a little snippet, may or may not be true, but what she pointed out is that, and this I think goes with other groups too, she said people often understand we can't defend ourselves. And I said, what people don't understand is that we actually go through a lot all the time. We're pretty good at coping with changes in some way. So I think it's a caveat. We shouldn't assume that when we don't have data that people are crumbling, even if they're starting out. A lot of people who have a lot of disadvantages get pretty good at coping with them. Obviously, there's a simple statement that has some caveats to it, but something to keep in mind. How does this, now those are what people report. How did this dovetail with what happens in a harder outcome? This was a great study that was led out of people from the UK. Initially, we were going to go through the literature as we did and look for suicide data. That wasn't coming out in the literature. So what they did is they went to any national, provincial, public health kind of data publications, so in one of the Canadian provinces, any country, and looked for trends. They had to have so many months prior to the pandemic, and they predicted what would have happened with suicides across, if the time trend had kept going with taking account seasonality and so forth. And what they found in short, that's what I just told you, what they found in short is that they looked at 21 countries, which have more than 21 data points. There are a few Canadian provinces, Quebec wasn't in this. But actually, for the first 6 to 8 months of the pandemic, suicides were either stable or went down across the board in every single place they looked. And they've updated this, and this hasn't changed very much as we've gotten, I don't know if we're still in the pandemic, or deeper into the pandemic, it didn't change very much. That's just the force plot. So basically these are, the dots on this side means it went down, and the line is a 95% confidence interval, the estimates. But they didn't have this, preordained, and actually it wasn't preordained that it was going to happen. People were saying it was happening and telling our population this, and it wasn't happening. That was wrong. So I guess in conclusion, there's no doubt that individual people suffered a lot in the pandemic. But in aggregate, I think this is an alarm or a warning to some of our colleagues out there too, that we're going out and talking without data, and what we tend in all of our specialty, I think if we're honest, we evaluate some of the burden and also what we're able to do about it. And there was rampant statements being made with definitiveness, and they weren't true. So I think people on an average or in the aggregate do cope, and they came together. And I think it discounts some of the really robust things that people did. Communities came together, online groups formed, people helped each other out. And again, this doesn't take away from individuals and where people suffered. Some people had better experiences. Other people thrive through supporting others. So I think we need to keep that in mind. There were some differences that are important to follow up on, and there were gaps where we didn't have data on some groups. But again, I think we should not, as I pointed out earlier in the talk, we shouldn't assume that certain groups did poorly because we don't have data for them. And then this one pretty much looked at one time point into the pandemic and hasn't followed it across. So this is turned into, again, very naively thought at the beginning, how much data would there be to look at? The team's gone through 160,000 studies and reviewed them. It's been, they're very good at processing in large. I'm ready for this to end because I'd like to get back to our normal business in some ways, but it keeps going. But they've gone through a huge amount. That's a massive amount of data they've gone through. And done a tremendously good job with it. So we're updating this, and we have now about 255 cohorts. Instead, it's about double what we did before, and the change is one. And we have about over 400 cohorts that have followed people across the pandemic that we're going to synthesize. It's a lot of work. So the team's working on not just identifying which studies we can look at, extracting the data. We do everything in duplicate to make sure we get it right, because we all make mistakes. And then evaluating the quality and so forth. So, hopefully in the next months we'll wrap this up and finish it. Anecdotally, I don't think we're seeing a lot that's very different from what I've reported here today, even as we go across the cohort period and so forth. And so this is a fantastic group of people who just did an amazing job. So just been a ton of hard work, and they work together, support each other, and get a good job done, and have done great. They also had people throwing a lot of darts at us along the way. People weren't very happy with our findings in some way. So I think they've had to young people had to stand up and know they were doing a good job. And despite there's like 1.5 million hits on a BBC article about us, so people are very angry about it. But there you go. So it's been an interesting experience for them, and they've done fantastically well. So there you go. So thank you.
Thank you. So again, we're a few minutes over time, so if there are any burning questions, perhaps I can ask you, or Celeste, no, you. So I understand that the change in the increase in the measures of depression were quite much smaller than expected. But nonetheless, given the huge numbers of people involved, who affected, doesn't that still, represent some measure of a public health crisis at the mental health level, or you don't think so?
I mean, it depends. I think it depends how you look at it. So if you look at on the general population level, I think that most people are doing well. I can be comfortable with that. If you look at, say, who's coming to an ER, and there's been stuff like that, yeah, that could change it dramatically. If you think about it in the population, you start doing the math here. If you look at, like, say, 2 to 3% of the people are coming for mental health care, and let's say that goes up some of these clicks, and you're seeing more people coming for care. So that, yeah, if you're working in a urgent mental health setting, the demand on those is going to get a lot higher.
Especially that our resources are already so limited.
Yeah, they're limited, and they're not always used particularly well. I mean, like there's a whole nother tough. If I came to you and asked you to treat me for rheumatoid arthritis, you wouldn't. But if you go to the way we distribute resources in our mental health system between private and public, and who gets care is not optimal. Mm-hmm. But yeah, it's going to put huge strains on that, right, when you do that. So there's two different issues here. What's the general population doing, and what might be happening in our limited care resources and that are already strapped, right?
Mm-hmm. Perhaps one last question.
Maybe I missed it. I sometimes do. What about comparing the population with long COVID with acute COVID, as far as depression and anxiety?
Long COVID and acute COVID. I don't think we have data on. I think the problem with long COVID, you need to know what's happening before they got it.
You'd expect a lot more mental problems in the people with long COVID. I mean, how discouraging can you get It's bound to have an effect on depression and anxiety.
Yeah, I imagine it would. We don't. I think you can assume it. You can assume it would. That most people with chronic diseases or bad chronic diseases have more mental health issues than others. So I think that's probably a safe assumption. But it's hard to actually measure that because we don't. Unless someone comes up, some of the American databases might be able to do that. Unless you have data that are actually kind of tracking some mental health ahead of time, and then you've got an exposure to long COVID and track it, then you could look at that. But I don't think anyone's done that. Yeah.
All right.
Thank you.
So thank you very much. So this brings this session to a close.
All right, so we'd like to conclude with a panel discussion. So I'd like to invite the panelists up to the podium here, and we'll check. Hopefully all the microphones are working. We have Amy Svatelis from the Fonds de Recherche en Santé du Québec. We have Heather Monroe-Bloom, Emerita Principal of McGill. Adriana Jarrett, who you heard earlier, who's going to be giving the patient voice. Marie Hertel, the chair of the Research Ethics Board, and Simon Rousseau, who comes from the Biobanque Québécoise de la COVID-19. And we were, as Marie was saying earlier, we're trying to figure out what do we want to present in the symposium and what voices do we want to hear and what ideas do we want to project. And one of the things that came up was, as Heather said in the opening remarks, SARS hit Toronto, it was unprepared, it was a disaster, and then when SARS-CoV-2 came, we were still unprepared. We still didn't have masks, we were still rationing N95 masks, and we didn't know what to do. So now, could there be some lessons learned? We're now recognizing from Don's presentation and Kiko's presentation also that the idea that you have an infectious disease and it just disappears after 3 days or 3 weeks or 3 months is naive. Things go on longer in time. And then you wonder, what are our tools to study the things that happen months to years later? Or do we just have kind of academic attention deficit disorder and we don't look beyond and we forget to look further? So I have questions for all of the panelists here, and I'm going to give priority to Adriana first. We heard her experience, and I just wanted to follow up on something she was describing. Who follows patients after they're cured? You don't know the Quebec system, but in the American system, does anybody follow patients after they are cured?
So for me, the health department technically does not follow you after you are cured. From the experiences I've heard of other people, they're done. Once you finish those last pills, they'll give you a certificate that says you've completed treatment if you're lucky, and they're done with you. I was very fortunate to have the nurse that I did have. I still keep in touch with her, but on a personal level, not on a medical level. So if I did have questions, I could reach out to her and she could talk to the health department about it, but that was no longer a role that she was required as of her job. The pulmonologist and the immunologist that I was seeing during treatment, I stuck with. I no longer need the immunologist after a little while, and then I continued to see my pulmonologist, but more so as routine checkups. It was only specific to the TB because I had TB, but yeah, there was no process in place, nothing put in place, no guide, like I said, for when I finished treatment on what to do. Anytime I had concerns, it was very much just, I don't know how to treat post-TB lungs. I just see lung damage and we'll try to treat that, but there is no clear answer for me.
Okay, thanks for that, Adriana. So Marie, I'd like to imagine that somebody recruited Adriana into a study. They got consent, and despite all the bad experience she had with doctors, she actually said, yes, you can have my blood, you can have my x-rays, you can study me going forward. I think some of the people in the room would like to know a little bit more about what are these data governance policies, why are there these rules that after X years you have to destroy the data, what happens if somebody retires but somebody else carries forward, what are the policies and how do they apply for chronic consequences of something that we see as a discrete event?
Yeah, that's a
Oh, is this on? It's an interesting question. In terms of policies, you will know that there are a number of layers of policies. So the question that I will answer is, what does the MUH-CREB apply in terms of policies, which is a different question from what you've asked. Sure. And you've got Law 5, remember the Loi sur la protection des renseignements de santé that's coming into effect in April, and that law will have a hard stop to store on data that's used for research without consent. Just a heads up. So the way that we function at the MUH-CREB is to work in the interest of the patient and creating value for the patient. And so I've seen a number of colleagues here with whom we worked during COVID, actually Simon, and we do not have a deadline on storage. Actually, we say, take away the 15, 25 years. We say you store as long as it's scientifically valuable and that you can sustain the resource. Because there are financial elements, and there are also, human resource elements. You have to keep these biobanks going. So in terms of long-term research, we have been fortunate that there is a trend to go forward with long-term banking. And I reach out here to Adriana. That was made possible because patients spoke out. I could also be, available for senior swimming pool time. 2008, was it 28? FRQ held a working group with patients because the government was hard on passing a law that would not allow biobanking. And there were a lot of patients involved, and it was the patient voice that got the government to change. And so we can do biobanking, but it has to be with consent. But there's no end on length of storage.
Okay. Can I follow up? Because I'm just curious, maybe the people in the room already know the answer to this, is what makes somebody a patient? Because I may be a patient of X, but I could be a control of Y. And how well is that delineated? I think in the late 70s, there were hepatitis cohorts in San Francisco, which taught us a lot about HIV in the 80s, but there were no HIV tests in 1979. So what is the definition of a patient in these sorts of situations?
Definition of a patient or definition of a control?
Yeah.
They come hand in hand. So what we have seen in multiple protocols is that it'll be a scientific determination, and it will be a control for this kind of disease. And so we see that a lot because, for example, if you wanted to get, I'll put out an example that I once saw, samples from the stomach from healthy controls to compare with patients. We would not usually allow that on the completely healthy. We would say, go for a patient undergoing a procedure clinically, take an extra bit of samples so they are controlled for this disease, but they may have another disease. So it will be, defined by medicine. Okay, thank you. Simon, you've been very involved in the biobanking initiative of the Biobank with COVID. The hard question is, what is the rate-limiting step that implies there's only one such step that's rate-limiting, but you're allowed to give us maybe 2 or 3.
Okay, thanks, because there were many. I'll start with the one that did not exist. At the onset of, and I'll talk about the experience from BQC19, the outset of the project, for once, funding wasn't a limited step. That was thanks to the vision from FRQS, Genome Quebec, the Public Health Agency of Canada, and MI4, who contributed. So this was a bit of a surprise to someone who's always looking for money. So we could see what were the other challenges. Biobanking is complex. You have the teams that will recruit a participant. You have consent. You have the legal aspect that ties the different institutions together. You have the ways you process a sample. You have those that want to access those samples. You have to make sure that they conform to the consent and to the management framework. So the challenge we faced was, while a lot of these existed in some part, they weren't tied together. And there's no one person that could actually be proficient in all of these things. We've had long conversations on Sunday afternoons following the onset of the pandemic, and there were great ideas I couldn't think of or weren't in part of the consent forms that we had. So the obstacle was the absence of these pre-existing links between the different nodes that are required to deliver the samples and the data from the participant all the way to the researchers that need them.
Can we do better next time?
We certainly would hope so. We've been working for the first 6 months of this year and trying to see how we could do better. We have a project that's called React Fast. And going through discussion with our stakeholders, one thing we noticed was that the legal agreements, and believe me, as a researcher, I don't wake up and say, yay, I'm going to be working on a new management framework or interinstitutional agreement, but these are the things that make a difference in this pipeline. Talking with the people that are involved with the ethics board and see how we can put in place these things ahead of time so that when it comes, we already have a system that can talk and that can be efficient in leveraging this. I also must say that we can do well in urban areas where there's hospitals, but I don't think we have a good outreach in communities, rural communities, and if we have something that's coming up, avian flu, for example, or zoonosis that may occur outside, then do we have the correct infrastructure to consent to capture the biosamples that are required to study the disease? So this is something I think we should think of as well.
Right. I'm going to turn to Amy from the FRQ. You've already heard a shout out that money was not rate limiting. And I'd just like to add another shout out for those who are unaware in Quebec, the physicians who are researchers are being, like subsidized or paid to do research and to think. And it's an incredible program. And whenever I tell people from outside of Quebec or from the United States, they just like choke. They can't believe what a great program that is. So those are two of the funding opportunities that the FRQ has provided. Are there other ways the FRQ can contribute?
I think, thanks for the shout out, and I'm going to do my own kind of shout out because I've always been very proud of the FRQS and the FRQ in general because we have the research centers, we have our networks, we have a system that's very excellent in Quebec in terms of supporting research and the patient voice that we've been trying to put forward, not only with the evaluation committees, but our programs. So those are some of the things that we often are able to use now as potential to put forward some ideas. For example, I mean, I could speak to any of the issues that were brought up in terms of patient follow-up and sustainable health, the santé durable. It's all about putting the patient first and looking at the patient in his context, right? So maybe speaking to something that Dr. Vin had in his presentation, he said, what's the study humans in the natural conditions to understand their diseases. I would say that's the sustainable health that we're going to look into. So what the FRQ does is much more than just their funding programs, but it's the advocacy behind the scenes as well in terms of data access, open access to science, it's many things, and ensuring that our evaluation processes include a respect and recognition of the collaborative aspect that's needed, because that's something that has come up with COVID is the interdisciplinary requirements and the collaborative requirements that were needed in terms of getting these groups together quickly. So I think that ensuring that those infrastructures are sustainable and the I in the MI4 that's the most important in my opinion is interdisciplinary, recognizing the people that can do that, but also recognizing that there are some fields that can't do that and will continue to work in the lab, but or on their own, but the interdisciplinary aspect is required for rapid reaction to some of these issues. Yeah.
Thanks. And another issue, it's a little bit more philosophical, but as you know, in Canada at the CIHR, there's research funding which is just based on the excellence of the project, and then there are institutes that create agendas and define areas to get behind. To what extent does the FRQ have these sort of two hats between this is excellent, we're going to get behind it, and this is a priority, and then what are the processes to define these priorities?
I think that's a great question because we have the foundation. So I've kind of compared the FRQS to my team last week of how we're like a house, So we have the foundations and the walls, and those are the scholarships programs, the career scholar programs. That's the building, And then we need the decorations. The research centers are part of the foundations. The networks are part of the foundations, and I think that one question that everybody asks me, and I've been asked twice today, what's going on with the networks? When's that coming out, Amy? And so it's coming out soon, just so you know. But that is one of the main reasons why the network program was rethought in terms of making sure that we stay up to date with what's going on in the research trends. We have the correct networks that are in place that will establish those links between each other. So that's definitely something that's going to be helpful and be more agile potentially in the future.
Thanks. All right, Heather, there's a tough question coming for you. There's a term that we really haven't heard all afternoon, and it's something that Heather pushed with the neuro, and there are opportunities. So where is open science in this kind of research, and how can we make our science more open, and what are the opportunities that we've overlooked here?
Well, thank you for the question, and let me first just say what a delight it is to be on the panel, and congratulations to the organizers of the day. It's been fantastic, and that we have everybody here as the sun is going down is very nice. So let me just speak to open science. When the Tannenbaum Open Science Institute was created at the Neuro about 6 years ago, there were some 60 countries in the world that had signed on to open science, including Canada, as a fundamental element of the way research would be conducted when it was publicly funded. At that time, there was not a single clinical research institute in any of those countries that had itself signed on to open science. There were, some of you will remember, NIH and NIMH episodic requests, sometimes demands that everything that was funded by them would be open science and so forth. Fact is, it was very sterile, very hard to get going. And as someone who spent 8 years of my life as a VP research at a large research-intensive university, where I had oversight of intellectual property agreements, I started out by learning how to do that, which was formidable. And then I realized that it was just an unproductive enterprise. I actually had a thought that never got implemented, that why don't we go for the windfalls, have a one-sentence piece of a contract that said if there's a windfall discovery that leads to a lot of money, which was the goal for all of that there'd be economic benefit back from this, then everybody could participate. But to burden every piece of research with complex intellectual property agreements when the hit rate for the economic side of it was so low made no sense. Long introduction. I'll get to your question, Marcel. So one of the silver linings, if you can put it that way, and there were a number of research silver linings to COVID, but one of the big global silver linings was that open science happened because everybody had something at stake. When the COVID pandemic hit, nobody really understood. I mean, this group would be a little different, but in general, people had no idea what was happening, why it was happening, or how to protect. And so the impetus and motivation to have a global connectivity on sharing everything one could in real time really broke that open. And I think there's been a sequelae of good practices and hygiene that's come out of that. But I do think that for us, the fact that MI4 was out in front of that creates a really good opportunity to keep building on the open science piece of that. And I think the fact that TOSI, the acronym for the Tannenbaum Open Science Institute at the Neuro, and MI4 are here and can collaborate together, just lifting up the platform of interdisciplinary and the kinds of research happening could be fantastic. May I just put one last PS in on one of the other questions?
Yes, you may.
And that is simply that, Joyce, you've got a however many decades career it is. I may have a very long one comparatively, and I would say the only thing in my, it was 1990 that I started in the organization of science and the creation of programs and so forth, that's made a difference to sustaining longitudinal research that's absolutely critical in this regard, and sustaining the whole range of clinical apparatus and practice linked to research has come about through advocacy. And you talked about patient advocacy, but the research community, especially the clinician science or clinical science interface, is just an incredibly powerful lobbying group. And it can be done in a third-party way that isn't just saying, give me personally money. And I say, again, that's a great opportunity for MI4 to help serve as a catalyst for that.
Thanks, Heather. We have one question at the podium. Don?
Yeah, thank you very much. Actually, I just want to preface or start this by going back to your original point, Dr. Monroe-Bloom, which is when SARS hit, we were not prepared. When SARS-CoV-2 hit, we weren't prepared. In between there, Ebola almost hit, we weren't prepared. And since then, Mpox hit, and we were kind of prepared. But we're a teaching hospital. And somewhere along the way, we lost the distinction, we've enforced the distinction that research is there and care is here, and they're two different avenues. But as a teaching hospital, that isn't our mantra, right? Our mantra is that research is care and care is research. So Marie, and again, I'm not putting you on the spot because we've had conversations about trying to get our protocols sometimes till 10, 11 o'clock at night approved. But how do we change this? Because 20, 30 years past, we're going to find ourselves saying, this is a hospital that does research sometimes on the side in that other building. Whereas what we really should be doing is using a model, I think, where every patient that comes to our hospital teaches us a lesson. It may be a lesson to, Professor, I was going to say Professor X, but not the X-Men, like Dr. X and Professor Y. It may be useful now, it may be useful down the road. Maybe somewhere down the road, we're going to have 4 or 5 more people who have M. bovis and aspergillosis, and we're going to try to understand the mechanisms behind there. Is there a way that we can change the paradigm?
I'm going to direct this to Marie because I'm really curious about this. I've heard this for at least 2 decades, that every patient at the MUHC will be a research subject. And it does seem from my undergraduate philosophy that sounds kind of like compelled or under duress, and there could be research, there could also be patient communities where we haven't consulted the community as well. So is this something that is philosophical, or is this something that is actually, based on legal definitions of consent and things that do or do not govern his proposal?
Actually, the law supports it.
Okay.
So the L4S, which will become Bill 15, and we'll see what happens at the end of that process, but it supports it. So, the mission of a SHU is clinical research as well as technology assessment and education. So the law supports it. So why is it not working? Maybe we could go for drinks. I think I think what people have to acknowledge is that the law requires REB approval of a protocol before you can recruit, and that's worldwide. Beyond that, there's so many things that can be integrated to facilitate the process, to streamline other aspects other than, get things moving. Some of you will know, and I see that Bruce Mazer has left, but we are trying to get the MUHC to work for a single centralized biobank. It probably won't happen in my career, but we're definitely working towards single departmental biobanks for several reasons. One of them being that you are more responsive to patient needs. Someone mentioned the neuro, they have a single biobank. And they are able to sequence DNA that's in their biobank to phone patients at home and say, hey, look, we've got this fancy clinical trial for your terrible disease. Do you want to come in and talk about it? So the children's is also moving in that direction fairly quickly. So I think that it's up to us to make the change, but the law supports it. And from an ethics perspective, I think we can definitely minimize the perception that ethics approval is such a big bump in the road. I think that's something that we can work on collectively. Some studies do go through quickly.
Great. Are there any other questions from the room at this point? Because if not, it's getting late, but I would like to ask every panelist if they have one last thought they'd leave with the room, starting with Adriana.
Oh, gosh. What's my one last thought? I didn't even get a chance to think.
We can pass and come back to if you want, or if you have something to share with us.
I think just kind of what I've been saying, it's important to remember that, as patients, we are people who like exist every day and have our regular daily lives, and so the medical part of it is such a separate part of it for us, even though as a doctor it feels like the main part of it. But being sick is not an identity that I wanted to hold, but it was an identity that I felt so strongly, and I didn't want it, right? But even though I was sick, I was still texting my friends. I was still like asking if someone wanted to go get lunch. I was still asking my professors questions. I was still existing in my life that I had before. And I think it's just important for doctors to remember that the sick part is not our entire life, and that we are people who have aspirations and wants, and that we know our bodies, right? And when patients tell you something is not right, we exist in these lives every day, and so an abnormal thing for us, like, we know that that's abnormal, and it's important for doctors to just check. Just check, just like reassure a patient, just let them go through the process of getting tested for something if they ask for it. Because we're so aware of our own bodies, we know when something is not right.
Thanks, Adriana. Marie, any last thoughts to leave with the room before you go for drinks with Don?
Sure. Anyone else? Well, maybe I'll echo Adriana's comments and say that we are launching on the, well, we have the MUHC Research Institute divide that we have to work with. But we are launching on the MUHC side to gather experience of research participants, as we do for patients. We have several surveys running at all times for patient experience, and we are in the new year starting to gather research participant experience, so that should be super interesting.
Great. Simon?
One of the slides that I like presenting most when I talk about BQC19 is actually the acknowledgement slide. On that slide, there's over 300 names. Many people in this room are actually named on that slide. Mary, you're there. And it's a testament to the energy and the commitment of everybody coming together. You know, for me, something I repeat quite often is that science, the human adventure. Well, our life is human, but we sometimes isolate this, and I really enjoy this. It's also a shout out to all the 6,000 participants that also contributed. So this is an amazing resources, and building on this vision, the pandemic broke some of the barrier. I got to talk directly with people I would not normally talk to as a basic scientist. I've talked with the Public Health Agency of Canada and engaged these different conversations that open different ways of thinking, different ways of doing things. So that's something I like us to keep doing as we move forward.
It's a very positive message. Amy, last message.
Thank you. I think it's an important message, because we talked all about this advocacy aspect, and as a research community as a whole, we have a really important role in addressing the gaslighting that does happen to patients, but also in addressing the importance of keeping that research community vibe together and showing that we have to combat disinformation, we have to change the paradigm on research is un fardeau pour le système, but it's more an advantage to the patients. And when I was working as a clinical research coordinator and recruiting patients and the nurses or other people who were like, no, on n'a pas le temps, I was like, no, it's an advantage, it's a right for the patient to be able to, for me to speak to them and give them that choice to have a research project, because it could be the choice sometimes between life and death. So I think it is really one of the major issues that we do need to work towards as a research community, is really combat that paradigm, switch it. It's not, time wasted, it's time well invested for the patients.
Thanks, Amy. Heather?
Well, I'll just follow up on the last three interventions and say that for me, one of the profound things was having the clinical, the research, and the patient come together today. I think it just was fantastic. And the other element is having time. Simone, to your comments, Amy, to yours, that it's so tough, especially I think coming out of COVID, all the adjustments, and then the world happening as it's happening right now, to take an afternoon like this and sit together and exchange. And it's enormously rich. There's no substitute for doing it. And when we feel we don't have time to do it, we just have to say, we're addicted to something we shouldn't be addicted to. We need to engage in this way to really advance progress. But kudos, félicitations.
Great. Okay, thank you to the panelists, and we're going to call up Marie for some concluding remarks.
C'est mon grand plaisir de clore ce symposium. J'ai beaucoup de remerciements à faire. Premièrement, I'd like to thank the speakers and the panelists for your thought-provoking presentations and for demonstrating the impact of your research. I would like to think that we should be proud that much of this work is being done at McGill, and there's so much more. And really, so thank you again for having taken the time to come and share with us. I'd like to thank the participants, and I asked myself, why do we thank participants? We thank people for taking the time to come. Thank you. I'd also like to thank the young participants. I hope some of this might be inspiring to you, and I hope that you're the next generation of people up on this stage, and I look forward to that. I'd obviously like to thank the MI4 team. So I see Kendra Tonkin up there she is the motor behind MI4. Tasha Allende, and do I see Philip somewhere? So Philip is somewhere. So thank you. There's Philip. Yeah. A big round of applause. And really, last but not least, I'd like to thank University Advancement and the MUHC Foundation for your unwavering support of MI4. I would like to think that we showed you what we're able to do and that there's still so much more to do, and really thank you for your support in our endeavors. So I have the pleasure of announcing a few things that might be exciting to some of the researchers. So today, I think we are launching the 6th Seed Fund Grant Competition, the 6th MI4 Seed Fund Grant Competition. So you should look to the website. We will be holding what I think is one of the more successful, or among the many successful activities that MI4 holds. The Seed Fund Grant are really important. The RFA is up, and I think we'll be expecting, the deadline will be sometime in early December. So get your thinking hats on. You heard Marcel mention that our annual report is up. The English version of that is up. Please have a look at the annual report. You can see a lot of the different activities that we do in addition to the Seed Fund grants, and ask yourself where you could perhaps contribute to that. The French version is upcoming. Our next, I think, MI4 public event will be a Dragon's Den in the early year. For those of you who participated in the Dragon's Den, these are usually quite fun and exciting. So the next competition for the young investigators in the room, I think we will be speaking to you and hoping to hear from you what your big dreams would be for your next 5 years. So the next competition will focus around the early career investigators. And the last point I will make is you probably right now have received in your email inboxes a survey, and we're gathering your feedback on this event. And as I mentioned, this is really important for us to hear from you, because it really helps us understand the needs of the community, and it helps us bring to you what are valuable events. So with that, thank you very much for coming, and we look forward to seeing you again next year.